it is.
The cat shark egg right in the middle.
Yeah.
The little orthioid arms coming off of it.
Yeah.
I'm really imagining the kelp-like texture you talked about.
Oh, I know what the kelp-like texture is like now.
Yeah.
All right, that's great. I gotten quite a few grabs. Oh yeah, you could zoom in more too. Looks like there's a little mermaid person there.
Yeah.
All sorts of other little things. Awesome.
I want to blow up a bat for my wall.
Yeah. So you can turn the lasers back on.
Got it.
Cool.
And thank you again for everybody on the ship for being so patient with us. It's easy for us to say, pick up that, do this. And I just want to thank you.
Yeah.
Our pleasure.
Trying to make it work.
Yeah, the spine looks really cool of this bridge.
The cloud spongerette.
Bridge nav.
Yep. Take visitor. Abby, our current depth is 286 meters.
I'd also like to remind the science team that we do have some Niskins available if you want water samples.
This would actually be probably a good place to just grab one while we're hanging around the wall.
Sponge heaven.
Yeah.
Yep.
So ROV team, if you want
And there have been a few little corals towards them.
If you want to go for Niskin number 3, we can try that.
Turn on the porch light.
The hake. Showing off in front of that sponge.
Yes, correct.
Watching?
Yep. Oh,
it.
there, it popped. Okay.
Thank you.
Cool. And so that is sample 148, 148.
Yep, thank you.
I'm currently working on developing the assays for doing environmental DNA sequencing of the sponges. First thing we have to do is expand our voucher library. So that's what the sponge collection, one of the things the sponge collections will be used for. And then we'll be analyzing this water sample to see how many of the sponges we can detect in the water sample.
Yeah.
Hey, what's light on?
Oh, wow.
Bridge chef.
I always love how the fish can just hang on these vertical rocks. What are you hanging on with? You don't have opposable thumbs. Abby online is pointing out that she's now seeing some Pocoastra, which is demo sponge species. She also went across the top mid of the screen. It's between the sea star and the yellow Mycale. Form these white shells with a furry brown edge. There's a neat shot. There's a nice header coney.
Yeah.
I'm keeping my eye out for a good cluster of Acrocolystes. Jenny has asked to see a good photogenic cluster to get a zoom on one.
Sure.
Nice little line of rockfish sitting up here on top.
Yeah, a lot of little fish.
Kurt says these are dark blotched rockfish.
So Abby is requesting in chat, if we see something that looks, a sponge that looks like a white plate, she'd like to get a zoom on it. So.
All right, I will keep our eyes peeled, and
Yes.
I will keep one eye on the chat.
Mm-hmm.
There's some little Acrocolystes. Abby was saying that yellow Michaelis looking sponge that we collected, there seemed to be more up here on top, and they appear to be growing, starting at that kind of little cup shape, and then expanding out into a much bigger kind of vase when they open up.
Bridgenet.
Can we get a zoom on that yellow cluster of sponge that's just coming
Zoom
to the
in, baby.
lasers? Oh, with the bonus cry noise.
Yeah.
So why is that one branch just sediment? Oh, look. Yeah, some of it's just sedimented over.
Mm-hmm. Just
Partially
little
dead.
portions of it. It's partially dead.
Yeah.
That's great, thanks.
Zoom out.
Can zoom out, yeah.
Hey, Meredith, can you hear me? I got knocked off for a while.
Yes, I can hear you, Liz.
Yeah, we can hear you.
Oh, we've got an octopus on the under the sponge on the right, crawling away off the bottom.
Uh,
Big one down below.
I didn't see it.
Yep, he's pretending to be a rock climbing down.
oh, it's gone now. Sharp eyes, though.
It gave itself away. It was already in motion.
Yep.
Why is that rock moving? Oh, octopus.
A rocktopus.
Mm-hmm. I keep seeing these little finger sponges that we collected one of earlier, and as they stick up, I've got black corals on the brain, and I keep thinking I'm seeing little black coral fins, and then going, nope, too shallow, and also sponge. Abby is saying that on sponge reefs, there are so many of these glass sponges kind of compacted together that it's hard to count individuals, so they count oscular density instead. Just how many openings there are pointed to camera.
You want to keep looking at the steep sections to the west of us. Follow this, because I think it flattens out.
Yeah, let's not go up into the flat so we'll keep looking more laterally. Stay in the steep stuff.
Sorry, I was just going to follow. I didn't know if they want to go up to the... So.
Following that ridge.
Yeah. Oh, it looks like there's more to go on the ridge.
We go all the way up to the rim and then kind of work our way along.
Like the better side of.
Pick the side with the most sponges.
It looks steepest on the west side of it.
Mm-hmm. Excellent.
too.
That little fish at the bottom of the screen looks like it's laying down to go to sleep. It's all tipped over
Yeah.
in that little crack. We do see differences in fish behavior between night and day. In the daytime, they'll often move more and get around, and at night, they really are asleep, even without the same kind of daylight cues that they would be getting more shallow.
Yeah, I was gonna ask, even with fish that stay kind of along the bottom, they show
Yeah,
daily patterns.
they still respond to the diet, like the daily pattern.
Huh. Oh, this is a cool little outcropping.
Yeah.
Lots of things hanging onto the edge.
Yeah, a nice little ledge system for stuff to tuck in underneath. We zoom in on, there is a brown ball that is just the same color as the sediment that is just to the left of the lasers right now. Lasers are just underneath it. Thank you.
Oh wow.
It is a sponge.
Yeah.
The oscula.
I think this is one. Yeah, I think this is one that may have already been collected today.
Yeah. We already got a brown
Yep.
sponge like that.
Yeah. Rancunia. They just look so cool when they're inflated underwater.
Yeah.
Little
These
moons.
these are the sponges we think are may have bioactive compounds in them.
Yeah.
Thank you for that zoom.
Oh wow.
Oh, wow. Another really big area.
Yeah. This one looks like it's partially dead.
Mm-hmm.
Look at all those little starfish legs poking out.
Yeah. Like every little tube has its own little set of arms sticking out of the top.
Yep.
That was great. Thanks. More sideways sleeping fish.
And another one of those ones that I think we already collected up at the top middle, just above.
Yeah, that kind of polymastia-like cushion.
Yes.
Yep.
For people at home watching, I know we're not talking much, but it seems to break the serenity of this a bit.
Yeah.
To talk too much. So we have about a half hour left on this dive, I think.
Yeah.
Would people say that's correct?
Mm-hmm.
So, and we have a few places left that we could put a collection. So Abby, pipe in if you see anything.
Yep.
We're just getting a handle.
Well, this was some nice Africalis there.
I'm still hoping to see that orange sponge.
Yep. When we collected it in 2016, it was just sticking out of the sediment with a flat top.
Is this one in the middle here another of the quesadilla ones?
Yes, I would call that a quesadilla. And we first thought that was a different species, but what we think it is now is a small one of a pretty common sponge in this area.
Yeah.
Now, I have to admit it looks more like a pita than a quesadilla, but
Yeah.
we sometimes name these things on the fly.
on the
Well,
fly and in the middle of the night.
when we are all hungry.
Yes. Yes. It's amazing how much, how food is prominent in our naming.
Bob, I'm gonna take your bubble to do.
Yep.
Gauge checks.
First person to see an orange sponge wins a prize.
What is the prize?
Orange sponge.
To be TBA, but it will be good.
A good prize. All right.
And it probably will involve food. I should tell you that I have my Nautilus mug out with me
Oh, yeah.
here on my desk to make me feel more like I really at sea.
You could also try
I
wobbling back and forth a bit.
have my Nautilus mug out, but I'm not feeling like I'm out sea because of the coyote jamboree going on down the hill for me right now.
Yeah, that's a real mood killer.
I was going to put my Nautilus shirt on, but I thought maybe that was a little too much. So what area will you be going to next year?
Next year is Hawaii.
Oh yeah.
Yeah, there's going to be another expedition in Papahanaumokuakea, isn't there?
I'm not sure. I'd imagine.
That's such a beautiful area. And you were there last year too, weren't you?
Last year?
We were there 2 years ago. Yeah.
2 years ago.
2018. Last year they were in American Samoa and the Musician Seamount. I track all of this with eDNA. Oh, look at that scrape. Or channel.
Oh, I see. Huh.
Interesting.
Also, that starfish looks like a cookie. It's just like the perfect star shape.
Yeah.
Yeah, earlier on this cruise, we saw some starfish that weren't as serene. They looked like they were always doing starfish yoga. They were bending around themselves and stretching out.
Oh, yeah.
So,
Well, I think earlier on the dive today, we also saw starfish that were just like, looked like they were flopped over on top of a rock, which was pretty funny.
doing a Pilates stretch.
Yeah.
So once you leave here, you'll be headed down to Central California. Is that correct?
First, we're at the Davidson Seamount.
Yep.
It's a little north of that. That's off the coast of Oregon, right?
Yeah,
Davidson is in Monterey.
Oh, yeah, Monterey.
California.
And Davidson is a beautiful spot.
Mm-hmm. They're going to do Pioneer Canyon in Greater Farallones. And I hear rumors that they're going to visit the Octopus Garden again.
Yes, that's something to see. So if anybody's watching at home.
Oh, and the Whale Fall.
Keep an eye out, and they'll be moving to that interesting spot.
Yeah.
And then you'll hear Meredith and I back on possibly when we're down more in the Channel Islands area after Gulf of the Farallones and Davidson Seamount.
The cruise leg with the Channel Islands. Also, we'll be doing Santa Lucia Bank, just north of the main Channel Islands. Oh, see that.
Fish. Not very many sponges.
yeah. Looks like we're kind of on the top of this.
Mm-hmm.
So I don't know if you want to keep on try to get on one side of this contour or keep on going up to the top of this, the last 15 minutes or so. We'll just keep going ahead.
I think going ahead is fine.
Great. Thank you.
Down front here. It's moving.
It's interesting. We're seeing more fish and less sponges here.
It's like they're all just kind of sitting up here on top.
Yeah.
Sleep. It looks like there's a little more topography over here.
Got some X sponges.
Mm-hmm. With some fish tucked inside.
Yeah, a little seabastomas, I think, maybe. Can't really see. Meredith, you want to take a bottle, right, at the end of this dive before
Yes,
we
we'll take
go up?
one of the last bottles. Yep. Steve is recommending that we, depending on how many Niskins are left, we pop at least 2 just to make sure there's plenty of water. That would be fine. We have 3 Niskins left, so yeah, we can, before we leave the seafloor, just pop 2 of them, and I'll use them as replicates. We can eel pout.
And another, maybe rosethorn. As I said before, there's lots of little subastomas, red ones with white dots. It's hard to tell them apart, or it's hard for people like me to tell them apart. Tom Leidig and Erica Frew and Kurt Whitmire are so much better at fish and probably could tell them apart. And they'll be back on identifying fish once we're down in the Southern California region.
Mm-hmm. So some of us will be joining through the science chat for the next 2 legs as well.
What's on there like this?
Just not in the headset.
It takes a village to do these research cruises, but we are so grateful to be able to participate in these.
Absolutely. Not the same as being out on the ship, but it's great to be able to actually talk with everyone on the ship directly.
You probably just popped the disc. Should probably tell the people that, yeah, we don't have 220 meters. Huh? Mark's probably out there. Yes. Just tell him. Sorry. Hang on. Okay, go ahead.
There's a fat sea star.
Mm-hmm. Looked almost like it was supposed to have 5 arms and grew an extra.
Yeah.
The other night, there was a rastenaster that looked like it had had every other arm lost.
By the number of arms that are partially missing or missing on sea stars, you can tell they have a rough life.
Mm-hmm. Well, that is a big sponge skeleton. With spot prawns under it.
Ah, yes.
That's actually the first spot prawn I've seen so far. Sitting up here on top.
Niskin bottle?
Yep, let's go ahead and we were going to pop 2 so that there will be a replicate.
Okay.
Yep.
So Niskins open are 4, 5, and 6.
Torchlight.
Yep.
So for those of you watching at home, these are plastic tubes with
Not yet. Yep.
All right, that's 4.
Challenge. Two corners there.
Yeah. Yep. It popped. Good.
Yep. All right, that's Niskins 4 and 5.
We're doing 6 too?
Yeah, might as well. We can use it for the extra volume for POM. So Niskins 4, 5, and 6
Good.
are going to be that sample. And we'll do replicate eDNA filtering.
Yep. Replicates are nice, and we don't always get them from working this deep.
Off.
Yep.
Well, that was a great dive.
Yep.
Okay.
Yeah, several of us were watching from downstairs at Preston how amazing some of those sponges were. They kind of look like melted candles in the past couple hours.
You're okay.
Yeah.
And we'll say goodbye to our rockfish friends here. The red-banded rockfish and Sebastianus rosethorn. And
Just a good recovery heading.
so we're headed toward the surface, right, guys?
Yep.
That's right.
Set up for it.
They're setting up right now.
Yeah, the bridge.
We're fairly shallow, so it should be just a few minutes, 15 minutes or so less before we're on the surface, and then deck team's already getting ready to get things out.
I was fascinated the other night because while they were recovering Argus, and the light seemed to attract a lot of little squids in. It was so pretty seeing them dance in the light there.
Yeah, I was wondering that. Is there a squid fishery out here? Because we were seeing some fishing boats with really bright lights off on the horizon at night.
I would think that those may be pot or longline fishermen. They tend to have their lights on all the time when they're out. Let's see if Erica's still on. No, but I don't think there's an active squid fishery up here.
Okay. Ready to start coming up?
Ready?
Do it.
Now it's time for spot the coolest thing in the blue water.
Yes. Have you ever gone blue water diving, Meredith?
I have not. I haven't had the opportunity.
I did a couple of times, and wow, is it great.
The shop I
And
dived
this
with
is
is headed to the.
headed where? I'm
Go
sorry,
ahead.
Meredith?
Oh, the shop I dive with is headed to the Galapagos next year, and we'll probably do some of that when they're out there.
For people at home, when I say blue water diving, it is instead of going to the bottom and looking at things like coral reefs on the bottom, you literally hang in the water column and focus your eyes near to you. And it's amazing what you can see just hanging in the water column. Sometimes you feel like you're a little bit like fish food. It can be a little disturbing at night, and you usually do this kind of blue water diving at night, but it's amazing when you just hang in the water column quietly what you can see.
You have to watch your depth really carefully.
Yes, because you can drift down without, it's very difficult to keep your perspective on depth and space when you're hanging there.
Mm-hmm. Well, we had some little squid at the bottom. I was kind of hoping they would follow us up, but I'm not seeing them.
I don't know. We seem pretty stable. I don't know if I want to monkey with it. Don't jinx it. Don't jinx it. No, I like it. Good. Not having to fight anything. I'm all right.
Are those
Our
metal
lucky
plates
data
that
log
are tied on? Go ahead.
our lucky data log team has a lot of samples to process once they get on deck.
Yes, hours and hours worth of work. Meredith, do you know if the plates on the front are ballast, and was that the plate they were talking about they could release?
I don't know. But
I think those are ballast, but I'm not sure.
Switch your USB now. Yeah.
Well, that was a very successful dive. We saw beautiful things, and by collecting a few samples of some of them, we are moving the understanding of those organisms forward. We'll learn a lot more about